Good morning. Welcome to video 7. This video focuses on cells, specifically organelles. 8 and 9 we might have a little bit of organelles in 8, but most of 8 and 9 will be on cell communication, cell membranes, active passive transport, cell signaling, that type of thing. Don't forget the free response. We typically have 1, maybe 2 free responses after each video that we would be working on. The free response we're working on after this video is free response 2018 number 6, and it's about cystic fibrosis. And again, for my students, if you go to Canvas, go to Files, then it's the one that says APFR referenced in YouTube videos. We're going to start at the middle of the cell, the nucleus, and kind of work our way out, so to speak. Okay, so what's the job of the nucleus? Well, it's to hold the DNA or contain the DNA. That's true, but why does DNA need to be held? If I'm a bacteria, I don't have a nucleus and I'm fine, so why do we need the nucleus? Well, there's a couple of reasons. One is DNase. We actually have enzymes that can destroy, or DNases. DNase enzymes can destroy DNA. Right? DNA polymerases build up DNA. DNases destroy them. We can have DNases in our cytosol. Cytosol, cell solution. So cytosol is technically just the liquidy part, sort of like the broth of the soup of life. And cytoplasm, your teacher might say that a lot, that's the liquidy part and the organelles inside. So floating around out here in the cytosol are DNase enzymes. They'll destroy DNA. That seems like a horrible plan. Why would you have that? Why is that logical? Well, the reason why is I can have a virus, a DNA-based virus, that infects my cells. And once it does, it kind of uncoats. And then if it has DNA present, my cells, they're made to read DNA. They'll just read it. They'll be like, all right. And they'll read the DNA, and that DNA will say, make more copies of me. And so our cells become a virus factory. So now, whenever the virus, if it's a DNA-based virus, there are also RNA-based viruses. But if a DNA-based virus goes inside, then hopefully DNase will destroy that virus. That means we need to somehow protect our own DNA. We either need to put chemical tags on there so that DNase doesn't recognize it, or we hide it inside of the nucleus. So the nucleus, it's better to say the nucleus protects DNA. So DNA can't safely leave the nucleus most of the time, unless we're going to shut down DNA. So like during cell division, mitosis for example, we need to have a way of destroying DNase or inhibiting DNase or something like that. Otherwise, when we start pulling this nucleus apart, we're screwed. Okay, so let's start here. You might remember my DNA is wrapped around histone proteins. Oh, by the way, I mentioned 2 reasons for having a nucleus. The other has to do with RNA modifications or RNA editing. And we'll get into that in the molecular genetics videos, but you might remember that before my RNA leaves, I have to put on a 5' cap, poly A tail, do splicing. Well, if I want to get all my splicing done before my RNA leaks out, I need a place to do the splicing. Otherwise, ribosomes would read it right along the way. So that might make a little bit more sense in just a second. So my DNA is wrapped around histones. Histones keep my DNA from getting tangled up into knots. Tangled DNA is unreadable DNA. For example, let's say that this was one side of my DNA, this is the other side. It's coiled up. If I want to read DNA later on when I'm doing transcription, I need to be able to unzip this DNA to read it, A, T, G, C, and then transcribe it into RNA language. Well, if my DNA is tied into a knot, then I'm going to have trouble accessing that. So if my DNA gets tied into knots, that's dangerous. So I wrap DNA around histones so that it doesn't get tied in knots. And you might also remember from gene regulation, when DNA is wrapped around histones, those genes tend to be off. When we acetylate those histones, we kind of loosen up our DNA. Now that DNA could be on and be read. So histones. I might have a gene right here. Again, this is kind of gene regulation stuff. I have regulatory sequences, a promoter, a terminator. So that's a gene. This is a different gene. This might code for muscle. This might code for bone. Or this might code for growth hormone or something like that. So in our example, we'll say it's some type of growth hormone. maybe for muscle and bone, like human growth hormone. This gene right here might have a protein on it that turns it off. So we'll get into repressors and inhibitors and things like that a little bit later on, but this gene is off. This gene is on. I might have transcription factors that are turning this on and everything else. So again, that's another topic for another video. But I'll do transcription. Transcription. So DNA codes for RNA, and then the RNA codes for proteins. Typically, not all RNA codes for proteins. But if I need to build a protein, let's say I'm going to build a protein and that protein is huge. That protein can't get out of the nucleus. We have these nuclear pores along here, and you can see those inside of this electron micrograph. Here's a nucleus. You can see it's got phospholipid bilayer, and then it's folded back over. That's why I draw mine with a phospholipid bilayer folded back over. So like, for example, that individual line right there is actually phospholipid bilayer. Technically, every other one of those tails is unsaturated and squiggly, but I was being lazy. So I have these nuclear pores, but if I build a protein that's really big, the protein can't get out. Almost like if I took all the parts and manufactured a car in this room, well, that's great, but now I can't get it through the door. So what that means is I have to build my car like out in the driveway. This analogy is getting odd. So what I want to do is I want to send all the information out of my room, out of the nucleus, out to the driveway. So DNA codes for protein. If I just read the DNA and build the protein right here, it's stuck in the nucleus. So I need to build my proteins out here. Well, if DNA leaks out here, DNase might destroy it. So what that means is I need to convert DNA language into RNA language. RNA language can safely leave where I can build this giant protein. Almost like if I have the English version for building a car here, and if it leaves the room, it gets destroyed. But I need to build my car down in the driveway, then I need to convert English into Spanish, and let's say Spanish language doesn't get destroyed. Then when it makes it down to my driveway, I can build the car out there. I don't know where it made sense in my mind. Okay, so we transcribe into RNA. And like I was saying before, there's some RNA modifications. We'll put on a 5' cap, a poly A tail, and we will splice out the introns and keep the expressed exons. We splice out the intervening introns and keep the expressed exons. So eventually, my piece of RNA, you might kind of draw it like this. So here's my strand, like that. And here I am kind of in action right here on this one. I've got a piece of RNA that I'm splicing out with the help of lysosomes. And so eventually I'll splice out all those blue pieces, those intervening introns. And now I've got my piece of RNA. So just the purples made it. The purples were the exons, the expressed exons. I put on a 5' cap. I put on a whole bunch of adenines, a poly A tail, and now this can leak out. So there you go. Now, what was this guy that looks like I always call that hamburger buns? That, by the way, says 5', just like in math class 5 and then a prime. So what is this kind of hamburger bun looking thing? Ribosomes. Ribosomes, their job is to read RNA and then find the matching tRNAs, transfer RNAs that are transferring their amino acids to join them in the right order to build a protein. So you might hear that ribosomes are the site of protein synthesis. Yeah, but more detailed, how do they do that? They read the piece of mRNA, messenger RNA, find the matching tRNAs to go in there that bring along their amino acids in the right order. If we get them all in the right order, that's primary structure, which folds into alpha helix and beta pleated sheets. that secondary structure, and then that folds to make a protein. Okay, so ribosomes might be free floating. If this grabs a piece of RNA and there's little signaling sequences that say, essentially, yeah, you're going to build a protein. Let's say this protein is green. So there it is, building the protein. And that protein's just going to go to work right here in the cytoplasm. Cool. That protein attaches. That protein now goes to work. Done. But sometimes that RNA leaks out, a ribosome grabs it, and there's a signaling sequence that says, hey, this protein needs to actually exit the cell. This is growth hormone, and I need to dump it into my bloodstream and send it to all my muscle and bone cells. So then that ribosome will go over and land on this squiggly stuff. Do you remember what that was? Yes, it's ER. Endoplasmic reticulum. Don't forget, if you're writing an essay on the AP Bio test and you want to abbreviate, say endoplasmic reticulum once, brackets ER, and now you can call it ER. When the ER has ribosomes on it, we call it rough ER. When that ER doesn't, we call it smooth ER. Smooth ER still has enzymes on the surface, but the enzymes are super, super small compared to ribosomes. Ribosomes are large. So under a microscope, I can see my ER covered in ribosomes. In this picture of my nucleus, where you can see the nuclear pores, you can even see those dots on the edge. Those dots on the edge are ribosomes. You'll also notice that the nucleus becomes the ER, or you could even say differently, the ER becomes the nucleus. When it's time to build a nucleus, a bunch of ER just sort of like lines up in a circle, and that becomes the nucleus. So that's why my ER is a continuation of the nucleus, because it is. Okay, so now, let's say that the signal sequence on this piece of RNA that leaks out, it's got its 5' cap and its mini adenine tail, 150 maybe, 200 maybe, adenines. That ribosome is going to read the piece of mRNA, and then what it's going to do is take that protein and then package it into the ER. You might remember we had our video clip called The Inner Life of the Cell. If you wanted to re-Google it, it's called Inner Life of the Cell. And that was the one where we had this whole process going on. In the Inner Life of the Cell video, there we go. You can actually see a ribosome. It's grabbed a piece of RNA. So here's the ribosome, the two subunits. They just call it a small and a large subunit. And the ribosome has senses again, a signaling sequence that says, hey, we need you to land on the ER and squirt the protein into the ER. So it's reading the piece of mRNA, and as it does, it's squirting the amino acid chain, the proteins, into the inside of the ER. So once that happens, then part of the ER is going to pinch off. And so now I've got my protein in there. Again, that video is called Inner Life of a Cell. So let's say I build my blue protein. Now a piece of the ER pinches off kind of like a lava lamp. And I now have something that looks like this. A vesicle. My ER just got slightly smaller because I pinched off and I'm taking some of this protein with me. By the way, in this process with ribosomes, eukaryotic ribosomes are a little bit different from prokaryotic ribosomes. So, for example, eukaryotic ribosomes have a 60S. They measure sizes of ribosomes in Svedberg units. And it's basically, if I remember right, it's a measurement of when they spin them down, how heavy they are, whenever they do a centrifuge on them. And so this is 60S and this is 40S. In a prokaryotic cell, they're slightly smaller. It's, I want to say it's 50-30. And the important thing though is the ribosomes are slightly different. Because of that, we have a lot of antibiotics. Ones that you might have heard of, it'd be like tetracycline, Seems like there's one other that you might have heard of. I might have even written it down. Erythromycin. Both of those act on prokaryotic ribosomes, but they don't interfere much. Some of them do interfere a little bit, but they don't interfere much with eukaryotic ribosomes because eukaryotic ribosomes are different. So it's almost like I might write a virus that, I wouldn't, but I might write a virus that attacks Apple products, but not PC products. Yeah, an Android phone versus an Apple phone. They do the same things. They text, they get on the internet, you can watch YouTube, okay? But they have different software. So even though they do the same thing, prokaryotic and eukaryotic ribosomes both do the same thing. They read RNA and build protein. They might have different codes. And so I might be able to have a drug that could manipulate this ribosome, but not this one. Just like I might write a virus that affects Android, but not Apple devices. All right, so now I've made this protein and I ship it. And once some of that pinches off, they call it a vesicle. So I've got this vesicle, and that vesicle needs to somehow get to Golgi. And so that's where we saw in our video, you'll have kinesin. You take the word kinetic movement, and he's a protein, so he ends in in, kinesin. Here's another picture of kinesin coming up. So kinesin will grab that vesicle and walk along. Now that we've looked at muscle contraction, you'll notice that kinesin looks a whole lot like myosin, for those myosin heads that grab onto the actin for my muscles to contract. He's a close relative. They're both motor proteins, so it kind of makes sense. But that's kinesin. You might have heard of dynein. Kinesin carries stuff towards what we call the plus end of the microtubule. He's walking on a small tube, a microtubule. And plus, towards the plus end means basically from the middle to the edge of the cell typically. Dynein goes the opposite way. So kinesin, again, his name comes from kinetic. Dynein comes from dynamic or moving. Okay, so now we've got this guy, kinesin, that's helping us out. So he's carrying this vesicle along the way, and then it gets to this organelle. What's that? Golgi. Golgi was discovered before the 1900s. It was like late 1800s, I think. And it's named after a person, last name, Golgi. So Golgi complex, Golgi apparatus. And essentially what's happening here is ER is for packaging, and it will add some chemical tags along the way. But Golgi is where we mainly process chemical tags. So we kind of like chop up some of the chemical tags that ER put on there, and then we start adding our own. A lot of the chemical tags that we add as this protein migrates through the Golgi apparatus, a lot of the chemical tags are sugars. So maybe in this stack of the Golgi, I will add that sugar. They call it glycosylation. Remember, glyco, kind of like glycolysis, glycolysis. Glyco is sugar. So we end up adding these sugar tags. And so as we go from stack to stack, there's not three, there's typically way more than three. We might chemically manipulate this some. And we'll make a Abraham Lincoln looking modification here. So just adding some chemical tags. Those chemical tags are important because that tells this particular blue protein, I think we said it was growth hormone, it tells it where to go. So these chemical tags might say, hey, you're a receptor. So go up into the cell surface, in the cell membrane there, embed yourself and act as a receptor or a channel or something like that. Or these chemical tags might say, hey, no, do exocytosis, like get dumped into the bloodstream. Okay, so now this part kind of pinches off. So like right here, remember how we said that that membrane right there is phospholipid bilayer? That means this membrane's phospholipid bilayer. So when phospholipid bilayer bumps into phospholipid bilayer, they merge. So when that vesicle hits Golgi, two become one. And you might remember from class, I give an example of when you used to blow bubbles. You have a bubble in the air, a soap bubble, a bubble in the air, and another bubble. Whenever they touch each other, sometimes they just combine to make one big bubble. That's what's happening. So my ER just got a little bit smaller, and my Golgi just got a little bit bigger. That's okay, because we can manufacture more ER. You might even remember we had actual cell videos showing that. We won't see those today. And so now I ship this guy out. Some of Golgi pinches off. And then once again, I have a kinesin that helps me make it to the edge of the cell. Like this. And then when I get to the edge of the cell, that's phospholipid bilayer and that's phospholipid bilayer. So 2 will become 1. And now this particular protein, and it might have maintained some of its chemical tags, is now maybe dumped into my bloodstream. So let's say that this is the bloodstream. BS, that's for bloodstream. Okay, so now this particular growth hormone goes cycling around everywhere in my body. Well, then you have something like this. I've got a bone cell and a kidney cell. So when I look at that bone and kidney cell, you'll notice if I look at the shape of these sugars that were put on here, glycosylation, in the Golgi apparatus, you'll notice that it looks like that top hat, that kind of Abraham Lincoln top hat, is going to fit to this receptor on bone. So it'll look something like this maybe when it finally fits. You'll notice on the kidney cell, that one looks like it fits a sphere. That one looks like it fits something triangular. That might even be the same receptor. They might share that one in common. And then that one doesn't fit. That one's kind of unique also. So, or at least it's not supposed to. With my drawing, it might actually fit okay. But you get the idea. So what that means is I can take a hormone like growth hormone, dump it into my bloodstream. Blood goes pretty much everywhere in my body. Not necessarily the corneas of my eyes, for example, or the lens of my eyes. But it goes pretty much everywhere. But in this example, bone is a target cell. So bone will respond to this particular hormone. Kidney is not a target cell because this is now what we call a ligand that hits a receptor. And that ligand does not fit any of these receptors. So kidney cells will not respond to that. So that's why I could have a hormone that I dump into my bloodstream and it affects these 5 different types of cells, but not any other cells. It's because of receptors. And some of those receptors are put on there in Golgi. A specific example that I know does this, where that sugar helps it fit the receptor, is gonadotropin. Okay, so we got Golgi, we got ER, that's rough ER, that's smooth. Golgi again was sort of chemically tagging, usually with glycosylation, sugars, to sort of tell a protein where to go. Now really, I guess I should say, instead of telling it where to go, this is in the bloodstream, so it's going everywhere, but more like tell the protein who to interact with. But it can also tell it where to go as far as, are you going to get stuck in the membrane, or are you going to be actually sent out of the cell? Exocytosis, a condition of exiting the cell. Okay, on the AP Bio test, they ask about this a lot. Basically understanding that DNA codes for RNA. RNA is read by ribosomes. The RNA never leaves the cell. It's that RNA is converted into protein language, which is then packaged in ER, sent to Golgi, sent out of the cell. So they like to ask questions like, we put a radioactive label. Obviously, you've never done this in a high school lab. I don't know that you can use radioactive labeling in a high school setting. But if we radioactively label RNA, the RNA would basically go from here. Radioactive labeling just means we can see it. It's almost like it's fluorescing. Think of it like that, or if we could color it, so to speak. So we're now putting a radioactive label on RNA. The RNA will go from the nucleus out into the cytoplasm, and that's it. Then if we radioactively label the protein would be somewhere here in the cytoplasm, if it's a free-floating ribosome, or if we end up docking that ribosome and we ship it out of the cell, then the protein would go from the ER to the Golgi and then either get stuck in the cell membrane, or it ends up like leaving the cell totally through exocytosis. So they ask that often. Okay, The next one that you'll notice up here that we actually studied in the previous few videos was a mitochondrion and a chloroplast. So mitochondria, why was that inner membrane all folded? It gives us more surface area for the proton pumps and the ATP synthase. You might not be able to see those too well, but that's okay. With the chloroplast, same thing. We're increasing surface area for the electron transport chain and the photosystems that hold chlorophylls and stuff like that. So that's why this inner membrane is folded. Well, another topic that comes up on the AP Bio test now and then is this topic of endosymbiosis. So how did mitochondria come to be? Well, that inner membrane right there is phospholipid bilayer, the green one. The outer one, the brown one, is also phospholipid bilayer. Your teacher might have called, or we call that inner membrane the cristae, also that folded inner membrane. So it's phospholipid bilayer. Most other organelles that have phospholipid bilayer, like ER and Golgi, it's just a single layer. Ribosomes technically don't have a cell membrane around them. They're just rRNA, ribosomal RNA, and proteins stuck together. So even though it looks like it's got a phospholipid bilayer around it, it doesn't. That's important because what we'll say is like eukaryotic cells have membrane-bound organelles, and prokaryotes don't. So they have ribosomes, but ribosomes are not a membrane-bound organelle. This is also kind of interesting because ribosomes are made out of RNA and protein. What's the job of a ribosome? To read RNA and build protein. RNA world hypothesis. So, with mitochondria, the endosymbiotic theory is this. When we look inside of our mitochondria, it turns out it has some circular DNA. Now, in eukaryotes, our DNA is a spiral, and it's just a single long string. So again, each chromosome is a single long string. We have, for example, humans have 23 pairs of chromosomes, assuming that we are normal. So here we go. Here's what it would look like in humans. In bacteria, same thing. It's still double-stranded. It's still a spiral, but we just connect both ends. So, for example, if this is a strand of double helix human DNA, in bacteria, the ends join, and now it's circular. Again, this part right here is still double helix. Just one end joins the other. So they noticed the inside of mitochondria had circular DNA. They also noticed that inside of mitochondria, they also had their own ribosomes. Guess what kind of ribosomes? They weren't eukaryotic ribosomes. Remember, eukaryotics had different size subunits, 60-40. And bacteria were, I think, 50-30. So they had ribosomes that were like bacterial ribosomes. And then they have DNA that looks like bacterial DNA. And so the idea is that if I look at a bacterial cell, like in this picture right here, they don't have a nucleus and ER and Golgi and stuff. So inside of a bacterial cell, you pretty much have genetic material and ribosomes, and that's it. They also noticed the mitochondria had 2 phospholipid bilayers. So then they started thinking, okay, my immune system cells, white blood cells, like say phagocytes, their job is, if this is a bacterium and I'm a white blood cell, once I identify that this is an unknown thing, I'm like, this is not a Mr. Cotton cell. I check the glycoproteins and glycolipids on the outside and I say, I don't recognize this, so I'm going to eat it. So it'll do phagocytosis and it'll engulf. Well, whenever you engulf, you look like this. So this bacterium is being engulfed by the cell. And then as the cell engulfs, phagocytosis, that bacterium will now have another phospholipid bilayer surrounding it, just like mitochondria. So we think that mitochondria came from pre-existing bacteria. Again, at first you might say that's ridiculous, and you're like, well, you know, mitochondria has their own DNA. Oh, well, does it look like bacterial DNA? Yeah. Oh, they also have their own ribosomes. Well, yeah, but does it look like bacterial ribosomes? Yeah, it's the same as bacterial ribosomes. In fact, some of our medications that are good at avoiding our eukaryotic ribosomes, some of our antibiotics, and they're good at killing bacterial ribosomes. That's why it's a good antibiotic, a good anti-life. It's a good bacteria killer. They also destroy the ribosomes that are in our mitochondria because they're bacterial ribosomes, essentially. Why does it have two phospholipid bilayers around it? Well, maybe back in the day, a cell tried to do phagocytosis but was unsuccessful. So we just wrapped it up and now it's inside. And now it's dividing inside of us. By the way, when you watch mitochondria cells divide, they do binary fission. We don't. We do mitosis. Who does binary fission? Bacteria. So they call that the endosymbiotic theory. Endo, inside, symbiotic, symbiosis. Remember you've learned parasitism, mutualism, and all that. That's symbiosis, commensalism. So endosymbiotic theory was bacteria was engulfed but not broken down successfully. Same thing with chloroplasts. They also have their own circular DNA and their own ribosomes. So we think endosymbiotic theory also applies to chloroplasts. More details on how mitochondria and chloroplasts, how they work, are last week's videos. Videos 4, 5, and 6 cover details of those. So I'll leave it at that, with one exception. In all cells, all eukaryotic cells, we have mitochondria as far as we know. There might be some exception out there. But eukaryotic cells have mitochondria because the job of mitochondria is to convert food, like glucose, for example, into ATP. The job of the chloroplast is to convert sunlight into food. Again, like glucose. So the idea is this, whether you're taking a 9th grade test or an AP Bio test or something like that, the idea is that they might try to get you to say something like, plants have chloroplasts instead of mitochondria. That's not true. Plants have chloroplasts. Instead of that, animals don't, so we eat. So how do plants get their food? Photosynthesis. We can't do photosynthesis. So what do? We eat. That's how we get our food. But once I have my food, I send it through mitochondria to convert to ATP. Once a plant has food, same thing. It still uses mitochondria to convert that food to ATP. So the difference between an animal cell and a plant cell regarding chloroplast is they don't eat. They are autotrophs. They self-troph energy. They make their own food themselves. We are heterotrophs. We hetero get it from somewhere different, troph. So we get our energy from somewhere else. We eat stuff. But once we have food, we both use mitochondria to convert it into ATP. This guy is a lysosome, and this will actually kind of coincide with what we were just talking about with mitochondria and chloroplasts and the endosymbiotic theory. So some of these vesicles aren't destined for exocytosis. They become lysosomes. Lysosomes are full of proteins that are good at just breaking down stuff. So some of the general names, they'll have like proteases, protease, ace is an enzyme, prote sounds like protein, so they just generally kind of break down proteins. Or lipases, they break down lipids. So they'll be full of just some general lipases, proteases, stuff like that. And the lysosomes are in the inactive state, or they'll call them sometimes primary state, the primary lysosome, inactive. And these particular enzymes only work when the pH is pretty low. Low pH. Was low pH acidic or was it basic? It was acidic. A lot of times students want to say high pH is acidic. I don't know why. So we have these proton pumps. Remember, a proton is H+. We went through that whole thing with how a hydrogen ion is an H+, it's just a proton. So proton pumps will pump in H+. H+ is what makes an acid an acid. Things that give up H+ easily are acids. Things that receive them easily are bases. So essentially, proton pumps will pump H+ in there, making that environment inside more acidic. Well, when that happens, those enzymes actually become active. So we always think of acids as like destroying a protein, but proteins have a window of life. So there are enzymes in my stomach. My stomach pH, my stomach acid pH is somewhere around like 2. And I still have enzymes that work in my stomach. They actually don't work at neutral pH of 7. They work at pH of 2, 2.5 really well. So these particular enzymes in here are responsible for breaking stuff down. So what they'll do is I'll have inactive versions surround. Here's what was supposed to happen with this bacteria. He's a bacteria and he's sad because he's getting ready to meet his death. So we just engulfed him, phagocytosis, and now these lysosomes start surrounding. And what they'll do is they're going to join. That's phospholipid bilayer, that's phospholipid bilayer. So as they join, 2 become 1, 3, 4, 5, 6 become 1. And so now, because 2 become 1, 3 become 1, and so forth, now these enzymes are covering this bacteria, and they should be destroying that bacteria. Punch them in the mouth there. And then we can do exocytosis to barf out the bits if we want later. Quick picture of, and so many pictures that we see in class that we don't get to see on this video. But whenever we see like exocytosis, okay, that's in another picture file. Never mind, that'll be a future picture on a future video. So then we can get rid of those things that we don't need through exocytosis. In addition to that, we have other things like cilia and flagella. Cilia and flagella are structurally the same, it's just size. So if you have like one really big long tail or 2 or 3 really big long tails, we call those flagella, or flagellum is singular. Cilia is if you have a whole bunch of small ones. So it's almost like picture a really, 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 really long hair, and then picture like a short whisker. They're the same thing. At a microscopic level, if I cut that piece of hair down and it's only that big, and I cut this hair down and it's only that big, they're the same. It's the length that matters. So if I have a cell, pretend that now this is a cell. If it has tons of little projections on there, and they tend to move stuff along like this kind of, those are cilia. If instead I have like a big long tail, kind of like we see on a sperm, then we call that a flagellum. So bacteria have no membrane-bound organelles, prokaryotes. And they basically, again, like we said earlier, they have genetic material, different ribosomes. They can have flagella, they can have cilia, and then in addition, they also have a cell wall. So the idea about cell walls, in general, this is not perfect, but in general, if you don't have a skeleton, then you'll need a cell wall to hold yourself up. There are some differences, like a jellyfish, for example. But what is it that's holding me up? Why am I not just a pile of goo, like just a pile of cells on the ground? I have a skeleton, a bony skeleton that's holding me up. If I was a bee or a beetle, I might have an exoskeleton that's holding me. What about those trees outside? I can see them, you can't. They don't have a bony skeleton, so they have cell walls around them that give them structure. Now bacteria are single-celled. As far as we know, they are always only single-celled, even though they can communicate with each other, quorum sensing, but they have cell walls around them. Knowing the exact type of cell walls in AP Bio probably doesn't matter. In bacteria, they have peptidoglycan. Some of them also have another layer called lipopolysaccharide. So if you had patho, then if you have a cell membrane, and then in addition to that, you have a cell wall made of peptidoglycan, then we call you gram-positive. If you have a cell membrane, and then in addition to that, peptidoglycan and lipopolysaccharide, a fatty sugary layer, then we call you gram-negative. If you're a plant, your cell walls are made out of cellulose. If you're a fungus, your cell walls are made out of chitin. The important part is that like a bacterium, a plant, and a fungus will have cell walls made out of different components. That's why, like penicillin, for example, penicillin won't hurt a plant or a fungus, but it will hurt bacteria because penicillin attacks peptidoglycan, peptide bond proteins, glyc, sugar. So whenever I'm a bacteria and I'm building cell walls, I take some sugar and some protein and I complex them together. Penicillin interferes with that reaction. So now when a bacterium wants to divide, it can't because it can't make a cell wall. Well, plants have cell walls. Why doesn't penicillin hurt their cell walls? Their cell walls aren't made out of peptidoglycan. They're made out of cellulose. So it matters. So just knowing that a plant cell wall is different from a fungus, is different from bacteria, is probably fine for AP Bio. If you're going to be a science major, now you know exactly what's different between them. Next video, we're going to go back and look at a few things just for interest. And we've got just a little bit more on organelles, but then we'll be focusing most of our time on active and passive transport and receptors and cell signaling. I did want to show you, I have another meeting coming up in just a couple minutes, which is why I'm slightly rushed. But I did want to show you some examples of like fluorescent dye labeling and stuff like that. So this right here, what you're seeing is the Golgi apparatus. And that Golgi apparatus, like we were saying earlier, it sends out vesicles to the surface of the cell. So these are all vesicles traveling to the surface of the cell. And you can see red are the microtubules, and kind of the yellow, you can see them moving around. Those are kinesins. We've labeled the protein inside yellow. But you're basically seeing kinesin carrying a giant yellow cargo vesicle with it, and then the roads that he's walking on are labeled in red. So let me go back one more click so you can see that. So these microtubule roads are labeled in red. There we go, sorry. And now, if from above you were looking at me and had this giant vesicle full of bright yellow stuff, then you'd be able to see it. So that's what you were seeing right there, was kinesin doing their work. All right, so many other cool pictures and things to show you. We might start off with some of those in the next video if it seems like we have time, but if not, we got to move on because these are review videos, not the learn everything videos. So thank you. Oh, don't forget, 2018, number 6, give it a shot.